Welcome back to our third tutorial. In this video, we'll explore the intersection tab, which is designed to identify common target genes between one or more diseases and one or more drugs. Inputs can come from Excel files, the provided example datasets, or our built-in dataset. Let's start by exploring the file input field. For this example, you'll need two Excel files, one for diseases and one for drugs. The example dataset uses schizophrenia data from the DISC-ET database. The columns of EntrezID, gene symbols, and disease ID, or name, are mandatory. A small alteration was made for demonstration purposes. Some disease names appear as schizophrenia and schizophrenia 1 to illustrate how a file works when multiple drugs are included. The same requirements apply to the drug Excel file. Again, for demonstration, we've included two example fake drugs. Since the results are formatted the same way for both files and datasets, we'll start by selecting two diseases and two drugs from the built-in dataset and explore the results. To continue our example from the previous tutorial, we use schizophrenia and schizoaffective disorder as the diseases. For drugs, we selected clozapine, which is already used to treat schizophrenia, and isoclozapine, which is not currently used but is closely related to clozapine. There are three main tables. The first table shows disease targets, including both the total number of genes and the number of unique genes. This makes sense as multiple diseases can share some genes. Each gene includes a link to the Jaccard's database, a widely used resource for gene information. This provides details such as genomics, DNA location, proteins, and more. For each gene, ReDEx also identifies the corresponding protein. Links are provided to the UniProt database, a widely used resource for protein information. The second table displays drug targets, following the same logic as the disease targets table. Finally, the third table shows the common target genes between the selected diseases and drugs, following the same logic with links and individual gene details. Let's save the table to highlight an interesting feature. The disease-drug column shows which diseases and drugs each gene is associated with. For example, DTNBP1 appears in both diseases but only one of the two drugs, specifically clozapine. This is a helpful touch that provides users with complete information. Next, we have a Venn diagram showing the total number of disease targets, drug targets, common targets, and the percentage of overlap. Finally, two separate heat maps are generated, one showing the similarity between the selected diseases and another showing the similarity between the selected drugs, both calculated using the Jaccard index based on shared genes. That concludes our third tutorial. Thank you for watching.